Hello!
Hi there.
Welcome to Microbiology Journal Club. We nerd out big about all things small.
My name is Danny. In a previous life, I dropped out for my PhD in microbiology where I was infecting skin I grew with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC dedicated to aiding the public with the tools of biotech.
My name is Faz. My PhD is also in microbiology, and I've mostly worked on bacteria, but I've also worked since then as a research integrity specialist, and I'm currently working as an editor for a scientific journal.
Every week, we meet to talk about microbiology. Today, we're doing an overview of some of the coolest microbiology papers we've seen this week.
At the end of the month, we'll be selecting a paper to do a deep dive into, so make sure to message us at MicroTWAC if something has caught your fancy.
You can follow along with the papers we discuss in either week in our shared Zotero library linked in the doobly-doo below.
And we want to hear from you, so please use the comments or tweet at us at MicroTWAC. And boy, do we have a show for you today. First, we're talking about maybe a third way out of the SARS-CoV-2 pandemic and how we can avoid further lockdowns. Then we have a cool study on how magnetic tweezers can tell, on what magnetic tweezers can tell us about remdesivir and how we can make the next antiviral better. Not only that, we are looking at how an immune signature might give us a clue about how to defeat HIV. We also have papers on metagenomics. And also, finally, we've got a story about how a defunct antibiotic might make a comeback. Tune in to hear more. So, now we're going to roll on the rest of the show.
Yeah. Yeah, so the first one is this, it's a modeling paper. And I was looking back at some of the deep dives we did. So the last time we touched a modeling paper, it was when the B117
Yeah.
strain came out. And then we sort of talked about like how they have different categories. They make, you make this like computer model where you have different categories of people and they like can be infected and they can get convalescent, and then they have a lower case of being infected later. And then you play around with all these variables to try to figure out what might happen, right, given a certain set of circumstances. And so this one was published more recently. This is a similar model style paper, published a bit more recently, but we're including this idea that test and trace techniques can help identify people who are isolated and stop the spread of an infection. So like this is incorporated into the model now. They have like a parameter for like how much testing is being done, and assuming that people that do get tested and test positive will be like isolated. And then they also, I think, have some. Do they also have something about non-pharmaceutical inventions? They do have.
Yeah.
Yeah, they have ways of reducing, right, the amount of spread, whether that be, some sort of mask mandates or even vaccines, I think, are being taken into account in this mathematical model.
That's right. Yeah. So yeah, they talk about let's see. Because we often talk about like say two options: or you either eradicate the violent virus completely,
Hmm.
or we wait until there's population immunity. But this paper seems to stake out a middle ground there, a stable
Yeah.
equilibrium of just kind of almost like what the situation is for some of the countries that are coming out of the outbreak, where it's a stable equilibrium with low case numbers.
Yeah.
Um,
And I think it's right. So I I mean, in some ways, I guess it's by necessity, right? This is a very useful piece of information to have because, well, people don't want lockdowns anymore. People want to be doing different things, but having some recommendations still uh, that come from some maths, some cleverly thought out maths, can help inform some policy. So yeah, I think this model is, I guess all models, right, sort of have some recommendation that I think that they're motivated by when someone
mm,
starts to make them, right? They want to somehow comment on what's being done. And so here, yeah, like sorry, you were just at that
oh,
yeah, you were
which
just at
figure
that
am I?
scale, that figure with the scale. I thought that was a good one to just speak about.
Oh
Yeah,
yeah,
figure
this one.
one.
Yeah, this one. Yeah.
Yeah,
Sorry.
so right, so here like these are the things that the model is trying to balance, right? And so like I think everybody knows, or not everybody, well, if it everyone was talking about R number, right? Reproduction value, like that was all based up. That was all in the virus itself, right? Like we're saying, and when we talk about the variants, right? It's like, oh, more transmissible, that's going to be a higher reproductive value, right? Influx, I think they're talking about things like how many cases are popping up from people entering right de novo from your population. Travel, right, is like the thing that's going on right now. And then on the other side, you got to balance that with test and trace and isolate, right? Hygiene, which is the non-pharmaceutical inventions, and contact reduction. That's again that's similar to that's another non-pharmaceutical intervention. And then you also have like immunity is on that side as well. That could be natural immunity, but it's also vaccine-based immunity. And they model out this idea that as long as you're not overwhelming the one side, right? So like as long as the case numbers are, so case numbers go into reproduction value, right? Like the more case numbers there are, the faster things are spreading. And so they sort of model out something where as long as you're not overwhelming one side of this equation, then you can have case numbers, right? And a virus that's out there. And like, if you think about it, this is also really logical, right? Like, for example, like the flu, we have outbreaks of the flu every year, right? But it doesn't like overwhelm everything because it only happens at one point in time. I guess that's another important thing. But also because people do get vaccinated, right? And like that is the push for these things. And I think what this paper is doing is bringing some of those because it seems like because we're going to be living with coronavirus like for some time, I think it's trying to bring those other elements in, right, to give us like not such a like you must lock, like if you remember from that previous model that we looked at with the B117, like they were talking about the action. They were really looking at how lockdowns can affect things, right? How closing, how like these really drastic interventions can affect things. And so I think this model, you know, we're moving into this era of like, we have to live with it. We have to do, right? We have to do businesses and things alongside coronavirus. So like, let's add some of those other features in the model so we don't have to, so we have something that we can study, right? Like smaller interventions.
Right, yeah. And those smaller interventions are like so masking, social distancing, kind of having limitations on like having better airflow in certain places.
Yeah.
Yeah, and they kind of like try to like classify the mild, moderate, and strong. And
Yeah. So I think you're going to figure two.
yeah, and it's like trying to like find out where that fine line is, and
Yeah.
it's kind of yeah, it's an interesting thing. It's just trying to figure out like what constitutes like the moderate or mild, strong, like kind of what is the dichotomy? I mean, I guess trichotomy there between the mild, moderate, and strong. But
Yeah.
but I do think that it is an interesting like presents an interesting idea that like we will have to live with masks and kind of social distancing and even like perhaps remote work for some time because of COVID
Yeah.
and that is like and but I think feel like for a lot of people the whole masking and like the social distancing that feels like the lockdowns or even though like it
Yeah.
isn't the same
Yeah.
but it's
Yeah,
because
no, I
now
mean,
we can
it's not the same. I mean, I think for the countries that did go through like an actual shelter in place or like some sort of actual lockdown, like it's a lot different now.
yeah
And like yeah, like I think in this figure they are actually looking at how lockdowns can affect in these different regimes, but then they move on into the next figure after that and they show, okay, well, you can also have control, right, without doing lockdowns. And I guess they also say like, yeah, or yeah, you can also control these things without lockdowns. I think this is what this figure is trying to get us
yeah
to take away from.
Yeah, so we can still keep business open, but the problem is you still need to have I mean you still need like masks, vaccines
Yeah,
and like some level of social distancing there.
yeah,
And yeah, it's difficult to get those like implemented.
yeah,
Because
yeah.
now
And
people want to get back to normal, I guess.
I was gonna say like I think it's tough too because in some ways, as I said, I feel like there's like a very strong pragmatism that's like running
Mm.
through this paper,
Yeah.
in a way where it's like we in one of the previous news ones we looked at like zero COVID strategies are sort of the only way to ensure that like everybody in society is like kept safe from things like because of like autoimmune or because of like immunosuppressed individuals and things like these there will be vulnerable populations. And I think that this paper, unfortunately, it's like, well, like here's a realistic way in which we've seen
Yeah.
things roll out, and like we're not going to be able to like the people who are high risk, I think, for COVID, like they'll be living in this like a little bit of a heightened danger, right? Like for the entire period until there's a better understanding. I think like this understanding about booster shots and things like that. Yeah, how to really prevent serious disease in our most vulnerable. Yeah, that's the frontier, I think, in trying to figure out public
And
health
of course,
things.
like disease also will be happening to people who wouldn't be considered vulnerable, wouldn't consider themselves vulnerable in that time, and there's going to be still. It's tough because like elimination would be the ideal strategy,
Mm-hmm.
but it's but for a lot of places it seems that the public will is not there for it.
Yeah.
Uh,
Yeah. Like
so
even
it's
right, I was saying even in really successful countries that were trying to pursue a zero strategy, like New Zealand, like they're no longer pursuing that strategy because of the difficulties faced with with strains, but also like it's also based on behavior, right? I think we've said that
yeah
every time. It's like like it's kind of crazy. Like public health is this very complicated thing because like you're really looking at human behavior on aggregate and like the news affects
mm,
that, like how people are feeling, like it's totally in flux. Yeah.
yeah. I mean,
But
you can sense the like kind of like I can see like the gain of freedom with immunization and seeing like this thing like, hey, we could be free if everyone got immunized, and
yeah,
uh,
yeah. And no, as I said, like I think that this is important to remember about models, right? And like we're not
yeah,
diving deep into this one to try to question some of those things, but you know,
mm,
one thing you can say is like every model has like a perspective on the world, right? Like
yeah,
when we were looking at that one, I looked at our old episode, the B117 one. When we were
mm,
looking at that episode, like it was very influenced by this idea of like we need to lock down in light of a new variant because if we don't,
mm,
there's going to be deaths, right? It's like it's
yeah,
influenced by a thought, right? And then they try to bring together like some parameters to help characterize that a little bit better. And I think here it's like this is a model for the age that we're living in now, right? Where it's like it's a lot more complicated. We know that there's different, like there are ways to reduce contacts, right, with other interventions, and like this is being incorporated and giving us some recommendations. And being us being
yeah,
like policymakers, right? Because it's like really
mm,
you do what you do. You should like try to be as safe as possible, I think, right? Like getting vaccinated is increasing your personal safety, right? Wearing a mask is
mm,
increasing your personal safety. But I mean, it also increases safety in this respect, right? On aggregate, right? Many people behaving in certain ways changes the amount of available contacts, and it's just a probability game.
yeah. So that so now I'd like to talk about the next paper, which is something that was quite interesting for me. So it's titled Inhibition of SARS-CoV-2 Polymerase by Nucleotide Analogues from a Single Molecule Perspective.
Mm.
Uh, so this is a paper about antivirals. So
Yeah.
so you've talked a bit about them on the
Yeah.
show. Quite a lot of the main ones like favipiravir, remdesivir, and last week molnupiravir. They're
Hmm.
all nucleoside analogs. They all impersonate a little section of the SARS-CoV-2 genome, the RNA. So the idea is that they get incorporated into the RNA and then cause the RNA polymerase, the thing that replicates SARS-CoV-2 genome, to have like a mental breakdown and just not replicate the genome properly or incorporate mistakes. And this
Yeah.
paper tries to delve into that in a bit more detail because we know like theoretically how it works. We don't get to see it in action. We don't get to understand what is going on in this process. And in this paper, they do something. They create this. They use this thing called magnetic tweezers, which are fascinating to me.
Yeah.
Um,
I think when we
so
covered the Moldunivir, the last
molnupiravir.
Molnupiravir.
Sorry, I got a slight cold.
It's like some sort of like Marvel villain's name. But when we covered that one, they did try to get at that mechanism at the level of like looking at what was happening to the DNA. But
Mm,
here, I think they're almost getting at even deeper into that mechanism, right? Being like, what are the forces acting on the the enzyme as it
mm.
incorporates this nucleoside analog?
I think that this is quite an interesting technique because what they do is they take a strand of RNA and they add like a magnetic bead onto one end and have like a glass like surface on the other, and then
Yep.
they apply a magnetic field to like kind of to like stretch out the RNA strand, and they keep this like steady force on it,
Hmm.
and then they like add your RNA polymerase to build up a double strand. And the thing about double strand RNA, which I've just learned about today,
Yep.
is that it's shorter than single strand RNA. So as the RNA
Right.
polymerase creates that second strand, the total length of the strand like gets shorter and shorter,
Right. That's
and
because
you
it's
can
getting
actually
wound, right? The winding.
yeah
Yeah. So it's like, I guess we don't really talk too much about these physics, but like, right, the base pairs they stack, right? They wanna stick together, and so
yeah
in that double-stranded, that's you get a helix, just like in DNA. Yeah.
yeah exactly like DNA. So you imagine like a ladder is taller than it would be if you like started spinning it around,
Yes.
started
Yes.
twisting it. So
Yes. Yes.
yeah. Uh,
Oh,
so if you
interesting.
want
Yeah.
to,
So
yeah,
so it's like a readout of they can read out on the force, right? The incorporation of nucleotides into that second strand.
yeah, they
Mhm.
can do it in like real time. So you can
Mhm.
actually like measure like how many strands are being incorporated uh like per minute or per second, and like you can
Oh
see
yeah,
like
yeah.
it, and
Because
the
if
interest,
they
yeah,
if they didn't have the readout of force and thing, then they'd have to stop the reaction every time and just like read the right, like see the length of RNAs that are being created. So like this, yeah, this is the real time. I see that now. Cool.
yeah, and the cool thing about this is that because SARS-CoV-2 has proofreading activity, it means that when like SARS-CoV-2 like when the polymerase like makes a mistake, you
Hmm.
can see it backtrack on itself or like pause and be like, oh, what am I going to do now? So
Right.
so like on this little graph here, you can see like these long pauses that happen occasionally for when something messes up, and
Yeah.
that's
And I think that
really like
from what we learned last time, it's like it's those pauses actually, right? That that the nucleoside analogs are dealing with. Like they could pause and then the strand could terminate, or they could pause and then something like non-canonical could get added in at that section.
yeah.
Yeah.
Um,
At least that's what I remember from what we talked last time. I don't know what it's this paper is saying.
yeah, I mean, yeah, I kind of only skimmed this a bit, but
Yeah.
I think what they found here was that like they tended to assume that remdesivir was a chain terminator. I think
Yeah.
that's something that we I think we said last week because during the
Hmm.
Molnupiravir paper there was a big thing of how it worked was different from remdesivir because remdesivir was
Yeah.
supposed to be like a chain terminator. Once it reaches that part, the polymerase just like chucks out the whole like. But actually, what this paper finds something different. It finds out what actually happens is that the polymerase backtracks and
Hmm.
then redoes everything before getting rid of the chain.
Hmm.
So it also like delays the so it kind of polymerase backtrack where it's yeah it goes back I think 30 bases and then tries to do everything,
Yeah.
and the thing is because of the way like previous techniques works, that looks like a chain termination, but it's actually something different. And
Hmm.
this is like I like this because of the tool, because it's an interesting tool to use to investigate how these nucleosides work, and you can figure out like they actually are doing to this RNA polymerase in real time.
Yeah. Yeah, I mean, it's also like the tool. I mean, they're not using it in this paper, but it's also an interesting tool just to look at RNA polymerases, right? You could like
Yeah,
do mutations in RNA polymerases and see how that affects its behavior. Because like they're very complicated and rather large enzymes as well. So
yeah.
there's lots of things that we don't know. Like, I mean, if people have been following along with all the spike stuff and you see these maps of spike and how big they are and how the mutations are these tiny little bits that affect, right, like their overall function, like polymerases are the same way. They're like these huge things. And so yeah, having this real-time readout seems super interesting and useful.
And even from like a bioengineering perspective, I'm just thinking about like how there's if they could do something like similar with DNA, there's this like measurement in lots of bioengineering circles called polymerase parts per second, where
Hmm.
they talk about how quickly if they got an engineered gene, how quickly that gene can be transcribed.
Yep.
Uh, and this could be a way of measuring that, because I think a lot of the
Oh,
methods
sure.
I
Yeah.
heard about like previously were quite indirect, but this is like a direct way to see how RNA gets extended. You can even look at there's a lot of cool things to be done with this technique. So I'm quite excited about it. And
Yeah.
yeah, this looks at the RNA polymerase of the coronavirus, but you can use other viruses as well, potentially.
Yeah.
Because the whole thing
You
about
don't have to
nucleus.
do RNA, you can do DNA as well, right? Like then
Yeah,
nothing about the system says that it has to be done with these particular molecules.
yeah. I think the whole thing about these nucleosides was that they're meant to be like broad spectrum.
Hmm.
And that means that you can actually test that by using different polymerases and
Mm.
figure out.
Mm.
And
Hmm.
also, you can try and figure out how to be more specific potentially as well.
Yep.
So I like this as a technique. Uh, it uses it uses magnets, and how do they work?
Totally. Yeah, no, really interesting. It also it reminds me of, yeah, I feel like biochemistry has gone like there's this like biophysics biochemistry interface now. Like when we were going through grad school, like there were optical tweezers were really hot. I don't know if we ever
Yeah,
talked about optical tweezers during any of
I
our episodes. I don't think we
don't think
have.
it. I don't. Yeah, I don't think they have. But they're
Yeah.
another really cool
Yeah.
thing.
But
I
you know
know. I
this
think we.
it's a weird
Yeah.
intersection, right? Because we're talking about force at the
Yeah.
scale of really really tiny things. And I mean, it's all physics. Even in the small scale, there's forces. But being able to measure them is giving us a new way of seeing, I guess, right? Like the dynamics and judging what's going on at this scale.
Well, yeah, I feel like in the 60s, what happened is a bunch of biologists went to a bunch of chemistry labs, stole some biochemists, and said, hey, do our work for us.
Yeah.
And in the mid 2000s, they did the same with physicists. They're going, hey, do you want to be like the 50th author on a Large Hadron Collider paper, or do you want to make some money? And they grabbed some physicists and they said, yeah, do our work for
Oh, they're
us.
not making money though. Make some
No, no,
publish
yeah,
a
it's
paper.
academic.
Get some street cred, some academic
Get some
cred.
street hub. Yeah, yeah. Just go ahead. Come, stay in academia. This is how we're gonna keep you in.
Yeah.
So yeah, we're seeing like a lot of fruits from that in terms of the way we analyze data now. These amazing new techniques.
Yeah.
So.
Yeah. Yeah,
Uh,
we definitely have seen the biophysics intersection when we did the biofilm dispersal one, right?
yeah.
Treating bacteria as if they were just like little rod things. Yeah, I would categorize that categorize this paper sort of in that realm as well. Okay.
Yeah. So next one is uh
Oh yeah.
oh wait, sorry, integrin activation is the one I did before that because I think there is a link in between, but integrin activation is an essential component of SARS-CoV-2 infection.
Yeah, yeah, this one is I think this is quite exciting because like we've talked a lot, well, if people have been following along, you see like there's so many different receptors SARS-CoV-2
Mm.
could potentially get at, and also like the severity of infection doesn't always correlate with the amount of ACE2 in the specific tissue. That was something that I think we call the callback episode there is the neuropilin one. Um,
Yes.
but here we're talking about another cell surface object that's found to be a co-receptor. So also important for getting in, but not for binding. It sort of maybe does some of the initiation in the endocytosis of these SARS-CoV-2s molecules. And so they look at, yeah, this other cell surface thing. It has like some flexibility. They know it binds to thing. It has associations with ACE2, and they know it moves. And they interrogate it using an inhibitor that prevents it
Yeah.
from doing this movement anymore. And they find when they use that inhibitor, SARS-CoV-2 doesn't get in as easy as it used to. Um,
Yeah.
yeah, so
Uh,
that lends credence to the fact that it's a co-inhibitor. And then they go, it's very biochemistry because
yeah.
then they go in and they have a whole bunch of different inhibitors and cofactors. If we go to the next figure 3, they start doing things with manganese and calcium ions, which I think
Yeah.
are useful for the particular motion of this integrin.
Yeah.
Yeah.
So I think it's like to do with like the cytoskeleton. So like it's almost like the cells got it like moving things internally as well as externally. So calcium activates it, almost like the same way that it works with muscle. I think.
Yeah.
I might be.
Yeah.
But
Absolutely.
yeah. So it's very much like the cell, and I think it's important for SARS-CoV not just for getting into cell, but also I think being transferred within the cell to do all the
Mhm.
cool things that needs to do there.
Well, I mean, you remember from the membrane, right? They replicate in a very specific area. So like they would have to traffic to specific areas, and that's also like quite important. Yeah,
Yeah.
I think that's the integrin is important. Is also the link between SARS-CoV-2 and the cytoskeleton, because like integrins, they are communicating with that structure, right? I think they're used in motility. They're used in a lot of different cell surface things that can remodel the cell shape.
Yeah, and it's a tough thing to like investigate because that this sort of thing is crucial to many cell processes.
Hmm.
So it's so I mean so shutting it down and finding out whether like the differences are due to you just shutting down the integrin or the entire system or just the SARS-CoV-2. It's an interesting sort of because it could be
Yeah,
because there is also this idea that like
you
it
just
might
broke
the
the
SARS-CoV
cell.
yeah, you
Yeah,
just broke the cell, and so even the SARS-CoV-2 can still bind to whatever it needs to bind to. The
right.
cell doesn't work, so
Hmm.
but I think they do navigate that in this paper because I think they 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 do some cool like mutation stuff to try and like figure it out. I think.
Yeah, like specific mutation. I mean, even yeah, I guess I didn't see like how they controlled with the inhibitors. Like you would expect that they might have something where, like for the general health of the cell, they could use that inhibitor and then
Yeah,
show that like the general health of the cell was still okay, and it was only this deficiency in bringing SARS-CoV-2 in. But yeah, I don't have any more detail than that from my skim.
no, that's yeah. I only skimmed it a little bit, but I think that
Yeah.
I
But
think that this is a nice model.
yeah, I think the reason why I want to highlight it is because it helps cut through the. So a lot of and I think we'll see it next as well. A lot of the tissue tropism and things that we see, they're done in vitro, and
Yeah,
it's also like they're done from this perspective of like ACE2 is the receptor. We see ACE2 on the cell type, so it must get in. Having more receptors and having co-receptors that are also important in the process of bringing SARS-CoV-2 in narrows down, gives us like can narrow down that field of places you can ask questions and maybe help people to make more judgments on whether or not like, oh, this is important because we know that it has the whole complement of these receptors, right, allowing it to be infected. So yeah, I think it's helpful from that perspective, just because there's so much, and we still don't know like a lot of things that SARS-CoV-2 is doing not in the lung. And I mean, I think
yeah,
that's really important as we enter this new age of SARS-CoV-2 as well. Like knowing some
yeah,
of the specific pathogenic mechanisms in places outside of the lung could help us illuminate stuff and correlate, right, with the symptoms that people might be having in long COVID or something like that, or like breakthrough
yeah.
infection. Like, are there long-term consequences? Not really sure, but we could begin to tell those stories, or maybe find the right. Maybe the story's already being told, right? It just hasn't been linked to the clinical significance yet.
Yeah. Which brings us to our next paper, which is highly efficient SARS-CoV-2 infection of human cardiomyocytes, spike protein-mediated cell fusion, and its inhibition.
Yeah, so really just another like long, another in the parade of different cell types that SARS-CoV-2 can infect.
Mm.
So cardiomyocytes, I thought it was kind of interesting because people talk about there's like if you get the for like for adolescents, there seem to be like some cardiac side effects when you get the vaccine, right, at some level. And then here, like actually
Yes.
SARS-CoV-2 itself, the infection may also drive something like that. So again, weighing harms. It's important to remember that like
Yeah.
vaccines are really quite helpful. Like SARS-CoV-2 is touching so many strange things in your body. It's better to just have a good vaccine, a good immune response to it, just to prevent who knows what's going on, right, when it gets into you.
Yeah. I think and I think early on in the outbreak, when ACE2 was found to be the binding, people were very panicked about the cardiovascular complications and like
Yes, yes.
endothelial system stuff. So
Yep.
I think this is still in keeping with that.
Yep.
Um,
Yeah, extending like yeah, going down that story. Although, right, we did cover part of the clotting phenomena, right? That was being mediated by some heparin perhaps, or like some sort of like some blood proteins rather than ACE2. But yeah, ACE2 is like super important in the endothelial and in the cardiovascular system. I mean, there are specific ACE2 inhibitors that are used, right, in cardiovascular health. And then another
Yeah,
reason I picked out this paper. Oh, go ahead. You want to
no, I
say?
was going to say, I remember like in the early outbreak, there was like a lot of concern over like what those things would actually be doing.
Yeah.
I know that like because I know there's a vaccine complication that I think was linked to either the vehicle rather than the spike protein, but there.
Mm.
I know like endothelial damage was like one of those symptoms people were very worried about at the beginning, and I think that's still something people are looking into.
Yeah. So this is again, we maybe got a bit off track. This is the cardiomyocyte. So this is the heart muscle cells
Yes,
that we're
heart
talking about
muscles.
in this particular one. And you know there have been some reports of right heart complications with SARS-CoV-2 infection, and so you know this is sort of the first step, right? It's in vitro, so like it's only the first step in sort of taking apart that pathogenic cycle. Yeah, if you go to the
Yeah,
PDF, like all the figures are right at the bottom. The other reason I chose is because
yeah,
they have pictures of cardiomyocytes and
yeah,
SARS-CoV-2 in them. Again, it's just cell culture, but like on page
yeah.
43, there's a SEM image of cardiomyocytes and SARS-CoV-2 all bound up to the surface of it. That's kind of interesting image for people
Yeah.
to see. Cardiomyocytes, I think
Okay.
we looked at them before. We may have even seen cardiomyocytes being infected before. But they're
Yeah.
very specific. They're because they're the muscle cells, they have these like structures in them, the myosin structures that do the contracting.
Yeah. They use human-induced pluripotent stem cell cardiomyocytes for this.
Hmm.
And I think one of the main questions because like these heart cells, they have like ACE2, but you need a furin to cleave that spike protein.
Yeah,
And the one we've mostly been focusing on is TMPRSS2, which we did like a show about a while ago.
yeah,
But that isn't present on the heart cells, so they were wondering, well, does that mean can stem cell get in?
yes.
And we have these like there are some like really cool images actually at the back of here.
Yeah.
So you get some like pictures of I think that's meant to be SARS-CoV-2, and
Yes, yes. Well, I mean, it's a cell culture, right? So cell culture, it's TEM, thin slices. There's not like ostensibly that cell culture is pure, right? They can do the controls and they can see that there's no viral-like particles. They add the SARS-CoV-2 and it appears inside of the cells.
yes, and they've got these electron micrographs that look like. Uh, I think is I don't know what the figure legends are, so I don't. They look like SARS-CoV-2 to me,
Yeah,
but I could be
it
completely
is.
wrong.
No, no, it is. It's supposed to be SARS-CoV-2 in
Okay,
this one. But yeah.
but yeah, it's yeah you can see it budding off the surface of these.
Hmm.
Uh, And I think there's also like this idea of fusion. So we've talked about like how spike protein, when it's expressed on the surface, can like help to fuse cells together as well.
Hmm.
And I think they've got like a really cool looking picture of like cells fusing together.
Yeah, so I think we talked about that a little bit in they had some like in respiratory syncytial virus, right? That's like I
Hmm.
think the classic thing where like cells fuse together. But like if you remember, when SARS-CoV-2 makes its spike, it has to go onto the surface of the membrane, and so like that's the membrane of the cell, and that's the molecule that's initiating these fusion events. So if cell membranes have them and they get close together, they can also fuse. Um, and again,
Yeah,
I don't
and
think
this.
that people have a good
Sorry.
we hadn't seen a good one that linked it all the way back to a clinical like
Hmm.
outcome. But it happens in cell culture, so like there's this idea that it could be happening in our bodies as well.
Yeah, I think like syncytia are like a symptom of some viruses because of the way the fusion protein works potentially.
Hmm.
But I think this is a really nice image because in green it shows like the SARS-CoV the spike protein,
Hmm.
and I think this is specifically the tail of the spike protein, and it's like she's showing it at the fusion point with another cell.
Yeah,
Um,
caught in the act.
yeah, that is like a cover image if I've ever seen one. Um,
Yeah, it's really pretty. I like the differential focus too on it. I mean, that's just like how it works,
yeah,
but like, yeah, they did a good
just
job.
Yeah, this if it was like the right actually no, yeah, it could still be a desktop background. I feel. Um,
But yeah, again, I think I really do present it more as a parade of the different, right, like the continuation of the different cell types that people are finding SARS-CoV-2 can get into. And again, there is evidence in clinical reports that like there are these clinical things that could be explained by this tissue tropism. And so again, it's good that we aren't going to the hospital and right like people aren't having, but there is this surface for complications that is still available, right, like with SARS-CoV-2 infections, as well as sort of previously unreported long-term effects. So
Yeah, and I think that brings us to the end of our SARS portion of the show. That brings us to the back half of the show where we start talking about other things.
yeah,
So
yeah. Wait, I just want to say with the SARS-CoV-2, I already feel like we're getting very like SARS-CoV-2 has become such an armchair. Like, first of all, we are armchair scientists.
yeah,
Like sitting
very much. I mean,
sitting in our
um,
homes and lobbing things from the peanut gallery at papers, trying to help people understand, right, like how it fits into the way people do science and stuff like that. I feel like with this batch, it's gotten even more so. Like the urgency of SARS-CoV-2 papers, the way that we were talking about them before, like really not knowing what's going on. It's now like we know all these things that are going, and we're finding these little details, these little detailed stories filling in gaps.
yeah, I mean, at the moment, what tends to be like, I think I've shifted a lot to looking at interesting technologies that have been
Hmm.
almost like prompted by SARS-CoV-2, like the magnetic tweezers paper,
Right.
or like, I mean, it's because now it's like I'm less like excited when I see like a new vaccine that or like a new I mean, and I guess the last big strain was the Delta strain, and
Yeah.
we are like if there's another strain, we'll probably get obsessed about that one as well.
Yeah,
Um,
absolutely. I
but
also, it's following
yeah,
media coverage. Like the media coverage hasn't been in the same way on those things. This week I tried to pick up like I was looking at the Novavax vaccine and I realized that like its phase 3 trial came out in September. And like we also like kind of like let that one slip a little bit by, but it's also like the approval seems like it's not going to even be through the FDA and it might be through like international. Like that might be some interesting news when that gets approved. And then again, vaccines for people under five to 12, and then under 5. Right, this is still a place that research hasn't happened yet. But again, very
yeah,
clinical in that sense, and we've never really enjoyed the clinical papers, anyways. Like
no,
because
not yeah.
yeah,
They're not very interesting from a microbiologist. They're mostly like looking at statistics, hoping that one number's big enough,
yeah.
and looking out for interesting side effects. Like I think there's one vaccine that makes you more likely to be murdered if you play with the statistics more long enough. But like,
Yeah. But anyway, I just wanted to give people like that little update on how I think we've been changing the way that we think about these SARS-CoV-2 papers, and like maybe people have seen that in our reporting. Again, tweet us, write us in the comments if you want to like us to cover something in specific. Like we want to help people understand what's going on,
yeah,
especially if there are things that aren't super clear about like papers, or like these like very strange details, right? Like that we're seeing in papers. That's also something that we'd love to dive into. But yeah, but other than that, like we usually have a good half now of the program talking about other things.
yeah. And next thing I'm going to talk about is HIV.
Hmm.
So the title of the paper is Monocyte-derived transcriptome signature indicates antibody-dependent cellular phagocytosis as a potential mechanism of vaccine-induced protection against HIV-1. That's a bit of a mouthful, but I
Yeah.
think to summarize what they're doing is there's been a couple of like vaccine trials for HIV, and some have done well, some haven't done. I mean, doing well is a relative thing because
Hmm.
it's very tough to make a vaccine against HIV-1 because it literally attacks the immune system.
Hmm.
So it attacks the CD4 helper cells, which is a key component of that
Yeah.
immune response. So it's very very hard to find out what an efficient immune response against HIV-1 is because we don't really have a model for that.
Hmm.
Um, except like in some of the studies, you get people with partial protection, And so
Oh, I was gonna say like
they
the efficient model I think is people talk about elite controllers, right?
yeah,
That's the
elite
only model,
controllers,
but it's
yeah.
like it's sort of like a specific genetic. People think there's a lot of like people say, what are the antibodies that those people make, right? Because those are the antibodies and that'd be very useful. Anyways,
Yeah,
that's sorry,
but
that's a digression.
they're all no. That's an important point because like there are some people who have genetic like resistances, like certain like mutation. I think
Mhm.
CCR5 that makes it harder for HIV to like to colonize to cause a disease. But
Yeah.
that isn't very useful for when you're trying to treat everyone because we're not at the point where gene therapy is there. I mean,
Hmm.
there are some people. I think there are some people who've had gene therapy treatments to cure their HIV, but it's not necessarily practical at this point. So
Have people really had that? I was not aware
oh yeah,
actually.
didn't you have the here at the Berlin patient? I think we talked about this, where he
Oh,
had a bone marrow transplant to
that's
make
right.
so
That's
his
right. The
bone
bone
marrow
marrow.
cells
We
were
did. Yeah, that was in a previous news episode. Yeah, the bone marrow.
yeah.
Okay. Yeah. Gotcha.
So it has happened, but we can't do that for everyone. So they're trying to figure out well what would be a good way to like have a control against this.
Yeah.
So so in this paper they like trawled back over data collected in previous vaccine studies, and they found that there was like this a specific transcriptome signature. So they just took like a bunch of like data from other places and looked at like the signature of like genes that tend to be expressed in people who control the
Hmm.
HIV outbreak
Uh-huh.
inside them. And then they tested this against the vaccine studies that failed or vaccine studies that didn't, and they found that so in this latest vaccine study, which was based on adenovirus 26, so I think very much in the neighborhood of I think that was the Johnson vaccine uses adenovirus 26.
Hmm. Hmm.
But yeah, and they that study didn't succeed, and they found so I think there's a little graph here. Uh, um, so this is like looking at like different vaccine studies and which ones succeeded and which ones didn't, and also whether they had the signature or not. I'm not so it's not the most tight of correlations because there is like one where they've got partial protection and no signature, uh,
Right.
but I think that this is like they're trying to figure something out here.
Yeah, I mean,
Um,
it's sort of this is the I mean, this is like some good work from like scientists who can dive into these large data sets, right?
yeah,
Like people are doing these experiments where they're collecting huge amounts of data, right? And they're going after it. But like what about synthesizing all those sets together to find something that other people have missed? And so like it might not be super clean because in a way, or a big source of variance here is everyone's done their experiments slightly differently here, right?
yeah,
But at the same time, like if they can find some good correlations, that may be like that little thing that someone missed. And here, I guess they're trying to inform future vaccine studies to try to say, here's an important biomarker you should be looking for that would show you a productive, like that would give a credence to a productive response.
yeah, exactly. I think this is a paper that couldn't really exist without open science. Open science is
Hmm.
like making people's data available to everybody to analyze so that you
Yeah.
can get it. I think this is quite an important thing to have, like papers like this that
Yeah.
pull what there is together and trying to find patterns. Um, yeah, and so what they kind of do is they reanalyze these studies and look and even break down into individuals and looking at who. So they basically go from one of the successful studies, I think RV144,
Hmm.
uh, and they like so they pull out like okay, does a signature in there or not? Again, this is a kind of data analysis which can be prone to like um, messing up a bit, so where you can overcorrect for things and
Hmm.
find things that
Hmm.
aren't necessarily real. Um, but the thing that made it interesting for me is that this signature they looked into like what it actually means, and I think
Hmm.
what it means is like that the important it kind of indicates something about the immune system because it represents the antibody dependent phagocytosis
Hmm.
by the immune system. So the idea is having high amounts of antibodies in the immune system will latch on to HIV and allow like all the innate immune responses to deal with it,
Hmm.
and which is a good thing considering the adaptive immune response is the main one that gets attacked by
Mm.
HIV.
Mm. Mm. Yeah, I mean,
So
actually, that like
yeah,
just hearing that takeaway from it, that does make me think that it's aligned with that idea of elite controllers having a specific antibody that can bind HIV. And once you get to that antibody, I mean, maybe it's like very hopeful, right? Like we don't know how to induce the immune response to it, but if we can, it will work. It will be protective.
Yeah, so it's kind of neat. I mean, it's
Mhm.
yeah, it has single cell transcriptomics, which is like my favorite technique. So that's another thing that
Oh yeah,
um
classic UMAP. Oh no, not
yeah,
UMAP
classic.
or TNSE. This one, but
Yeah,
the clustering algorithm that lets people see all the different types of cells based on their gene expression.
yeah. Unfortunately, eLife's zoom function is the worst thing ever. But I would zoom in, but then everything gets stretched out, and it's. But yeah,
Yep.
so any hope for HIV is good hope, I feel. Yeah, and now
Yeah.
next paper is a selective antibiotic for Lyme disease. So I like the story for this because Lyme disease, it's spread by ticks. It
Hmm.
produces like some very distinctive rashes afterwards, and nobody likes it, and it's on the rise because of global warming and ticks spreading into more areas that they wouldn't usually spread. So we we don't like in Conversely, ticks are the most grossest creatures on the planet. I feel. I
Yeah, they're
feel
pretty gross.
so, but what they did was they said, okay, well, let's try and find a new antibiotic. So they isolated some soil microorganisms, and they found that there was a compound highly selective against spirochetes, which
Yep.
is exciting. It's a new antibiotic, except
Hmm.
when they found it was actually hygromycin A, which is a very old antibiotic
Yeah.
that people had written off because it was pretty not very good against most diseases.
Hmm.
So, um, but apparently it works really well at spirochetes. So this is like a comeback story from like
Yeah.
a people wrote off hygromycin, but now they've found that it works really good against spirochetes, which I don't think they probably didn't test
Yeah,
against too
I
much.
also think that that's like such an interesting, it's such an interesting way to come to an older antibiotic because I think I'm not sure if we covered this explicitly, but it's quite common to have panels of like, repurpose things that need to be repurposed, right? And like
Yes.
they'll put those in like big libraries and then people will screen through these libraries of drugs that have already been approved or have already some use and like they need to be repurposed. But like here's something that like we did know about, but I guess never like made it into those sorts of screening libraries that we had to re-find it in nature to be like, oh yeah, this is actually important. We could use it.
Yeah, I guess it was discovered in the 1950s and it just didn't take, and now they've got it back, and yeah, it does something against was it DNA replication? I think maybe inhibits translation. So yeah,
Yes.
and
Yeah, so
so
ribosome stuff.
yeah, ribosome stuff. So yeah, stuff we could investigate with magnetic tweezers potentially. Um,
Yeah, I mean, we didn't talk about that, but like, yeah, you could put a magnetic bead at the end of a peptide even, and see
true.
what's going on. But yeah, but I don't think that and peptides they fold as they come out. So anyways, that's
Yeah,
I'm
it's
getting very
really good.
distracted now. I'm thinking about that.
Yeah, sorry.
But yeah, no, hygromycin B. I mean, yeah, I don't know what else to say about this except very funny way to find it, and it is always good to have something against Lyme disease. I mean, I would
Yeah,
like to see. I don't know what this is like. So Lyme disease is this pathogen is intracellular, right? It goes inside of your cells,
yeah.
um, and so it's already a just by the nature of it just having an antibiotic for it doesn't really mean that it's going to be super useful for the
Mm,
disease itself just because you're going to have to give like a really high amount. It's going to have to get into your cells in order to get to this spirochete that's living out inside of there. um, but yeah, it's still very
yeah,
promising. Always good to have another tool in the toolbox.
yeah. Next one. Biofilms can act as plasmid reserves in the absence of plasmid specific selection.
Yeah.
Uh,
So this is a really small a small paper, and again with the story of antibiotic resistance, a lot of or like when thinking about infections and antibiotic resistance, a big way that they're transmitted is through plasmids. Plasmids
mm,
have like the genes required to become resistant. And that could exist in nature or something like that. And in nature, bacteria live in groups often, not just
Hmm.
like single solo bacteria, and definitely not homogeneous colonies, or yeah, usually not.
Hmm.
And in this paper, they just look at a mutant actually that can't disperse biofilms as well, and they show that that mutant holds on to its plasmids better. So usually plasmids are selected against because you have to make like this extra piece of you have to make this extra piece of DNA inside of you. But you can imagine inside of a biofilm, maybe it's not a super competitive environment. You're with all your kin. So like people might keep around their plasmid a little bit more. And if you don't disperse around, then I guess that's the selection pressure that maybe helps these plasmids stay. Which
Yeah.
is just a strange dynamic. I think it's a really cool observation, and again, I think it's really important for people to be focusing not just on how bacteria live in our bodies, right? But when they cause disease, right? Like in that specific pathogenic interaction, but also like when they're just hanging out in the environment, because a lot of that hanging out is this could be in a hospital, right? Like these could be
Yeah.
like bacteria hanging out in the hospital. Like all it takes is like even though like the plasmid isn't being selected for, they're still like holding this memory of it right inside of this little like smudge of biofilm that lasted somewhere, and then that could like just start all of a sudden when the selection pressure emerges, start propagating throughout
Yeah.
the hospital. Yeah, these are the types of things that the the I don't know the attack surface for
Hmm.
humans, right, is like quite large, and understanding like these different mechanisms in bacteria is a way to maybe inform strategies to disrupt it. Maybe all this says is just clean everything, right? But like, but it's a specific
Yeah.
dynamic, right? Like that's emerging that biofilms hold on to plasmids.
Yeah, I think it's a because it's important because it also kind of speaks to like different roles bacteria have in the communities and
Hmm.
like how some of them like are metabolically active and can't, and like more likely to shed the plasma than others.
Yes,
Uh,
yes.
and it's quite and also like put like plasmas persisting in the environment and yeah, it's there's a lot of like cool little ideas in this paper. It's
It's
quite
very
short,
short. Yeah,
but
it really is just I think like the main thing is this observation, right? That like plasmids do persist better inside of biofilms, especially and when the biofilms don't like get dispersed, they persist even better, right? And again, you can think about that quite logically and say, well, yeah, like those non-dispersing biofilms, they're very static. They probably have uh, very difficult nutrient concentrations and oxygen concentrations to deal with, and so the cells are a bit more dormant. So they don't care that they have like this little piece of DNA around, as you said. Yeah.
which I think links back to a paper where we talked about last week about like uh, biofilm dispersal being very important. And so, if you want to get rid of those plasmids, dispersing biofilms, forcing the
Yeah.
bacteria into a position where they can't keep the plasmids around, could be very important. So, yeah,
Yeah, like you could I think we were talking about you can add in things that disperse, right? Biofilms, or you could
yeah,
have I mean, we've been talking about these like weird decoy bacteria that people can make, right? A bacteria
yeah,
that doesn't behave with the collective interest of the group, and it's just like, yeah, we love to disperse biofilms. When we grow,
yeah,
like we send out the signals that say let everyone, let's just like run away. And so yeah, there's like all these kind of interesting ways of manipulating bacterial ecology might end up someday being useful in a clinical sense.
sure, yeah. Next paper is recovery of strain-resolved genomes from human microbiome through an integration framework of single-cell genomics and metagenomics. So, yeah, this is very much a paper that I'm quite interested in because of single-cell genomics and metagenomics. Because metagenomics is like
Yeah.
it's you going out in the field with a bucket, putting all these genomes in, and they're mixed together, and you have to figure
Hmm.
out which genes come from which bacteria. And there are ways of doing that by like trying to look at species and related genes,
Yeah, and like
but also,
it's
yeah,
overlaps as well, right? Like they they
yeah,
have really good algorithms that look at all the different overlaps, and like hopefully you get enough overlaps. You're like, okay, this is its own thing and not like does not overlap with this other thing. But it gets complicated
yeah,
because you
because
can
even then you can yeah, you can
everything's
make
blended.
mistakes. You can, yeah, because sometimes you get very related bacteria genes can jump between different bacterial species,
Mm-hmm.
and you have plasmids doing the same thing, so it's very difficult. But then you got single cell genomics, where you can literally like take your sample, shake it up, and like put every single cell in its own little bubble, and you can sequence that genome.
Yeah.
The problem is that I mean, that's a great technique, but apparently like they don't get all the genes from it. What they do is that they get a couple of genes.
Mm-hmm.
But that's actually very useful if you couple it with metagenomics. So if you take your bucket sample and then you like take half of it into single cell and half of it into metagenomes, one bucket has all the genes, but you don't know what the order is. The other one has a few genes, but it's like you get
Wait,
all the important puzzle pieces.
I was gonna say maybe you want to scroll down to figure 1. I feel like they try to show what's going on here.
Yeah.
Yeah.
Yeah. So you got your like genes. So you got your like kind of single cell genomes, and they've got some fragments in there, and then you've got your metagenomes, which have lots more. And if you can use those single cell fragments and use them almost like a search algorithm to see what matches in the metagenome, and then from that you can build up a full genome from that.
Yeah, they call it binning, and if we cover more metagenome papers, like this term will come up a lot because it's like it's a specific strategy to get at the fact that you're dealing with all these tiny pieces everywhere, right? And you're trying to associate the tiny pieces together, right? So that they are in meaningful groups that relate something to the biology. And so single cell sequencing is great because we know that those things that, the sequence that comes out from one of those droplets has some biological meaning. It lives in a single cell. And that informs then this crazy shotgun approach of the metagenome where you just get tons of sequences from all over the place. But now we have like a little bit of a guide, right, to guide the binning of that giant shotgun.
Yeah, and it's really useful if you're studying plasmids and where they spread to in the community, because that single cell, you know that single cell has a plasmid in it if you can recover some genes. And suddenly now you can see if that plasmid is in other cells with maybe of a different species. So it's really useful in figuring that out. So I think this is a really neat.
It's super, I think this is super cool too. And again, like single cell, we've seen it being used in humans, right? Like where you can see all the different cells in a tissue, but like using it in the environment is also an amazing way. I think actually we have a paper in our potentials list that says it's about like doing, like studies with single cells to get at some like assigning metabolite function into some of these like cell categories. But just like very cool techniques that are given to us by single cell for sure.
Yeah, and I think that brings us to the final paper: decreased growth of wild soil microbes after 15 years of transplant-induced warming in a montane meadow.
Yeah, this is definitely like a total departure. I've been trying to like throw in, right? Like I always want to throw in some papers that are like a little bit outside of what maybe we always talk about in pathogenesis and health. This is just transplanting like cylinders of soil from one place into another place, and that other place has a slightly different climate, and they track the metabolism of the microbes in that different climate to show that they actually grow slower in this warmer environment. And the technique that they're using here is called stable isotope probing. We haven't talked about this before. Essentially what it means is they have some isotopes. I think they're using oxygen of some sort, right? You have different weights of oxygen out there in the world. And so they know that if the isotope gets incorporated, the amount of that isotope getting incorporated to the sample is a proxy of how much oxygen that sample is using. Right, so then they get this idea of growth rates as a whole, and they don't actually have to go in and like culture things and say like they're growing. They actually know like this chunk of soil is absorbing this isotope at this rate, and that's how they get the growth rates.
Yeah, that's really interesting. I mean, I like the idea of them just like taking a bunch of soil from one place and then putting it to another, and then monitoring it for like 15 years. That's like dedication, I think.
Absolutely, yeah. You get this really it's like such a nice long-term. I mean, one of the wonderful things that I've always liked about science is like you get these projects, right, of like intense concentration, right? Like the plan was made and they followed through with it. I think we've seen this in a lot of ecology style studies, right? Like they tried to grow like some like nutrient poor, right, microbe that like lives under the crust or whatever. It took them like years to grow it in a bioreactor. This is like more applied in the sense that they're using actual samples. They call it a mesocosm, right? Because it's like a medium-sized environment that they move from one place to another. Yeah,
Yeah,
and
and they keep everything intact as well, because I mean we did that entire paper about how important soil structure is. So so yeah,
yeah.
they are
They literally
observing
take the
microbes.
core out and they put the core into a similar meadow that is just in a slightly different climate, a warmer climate.
Yeah.
Yeah, I guess, one of the things that I just want to relate this to the way that we talk about models a lot, right? Like we talk about models of disease, right? And we're saying like, oh, like we try to like replicate, right, this specific disease process, right, in an animal, but we know that it's not the human, right? But we hope that there's parallels in between there. And like there's something to be said about this where it's like we know that like it's so totally artificial to take a cylinder of soil and stick it in another place, but like that temperature is different, right? And they try to maintain as much of the similarity as possible, and it becomes this model of climate change, right? That during
Yeah.
climate change, as environments warm, we will see slower microbial growth. And then, you have to take that the next step down and say, well, slower microbial growth, what does that mean? Like, what do these microbes do, right? Their metabolism in the environment, how is that impacting other animals, right? This doesn't talk about that complexity of this particular thing, but it extracts it out and gives us this model to study it.
Yeah, I mean, I know that there is like a lot of like interest in this kind of thing because where the topsoil is being depleted by agriculture and other sorts of things, and the idea of like what happens with global warming, what happens with I mean, how will this affect like food growth? Or I
Yeah.
mean, I guess another thing is like the carbon emissions as well. There's lots of little details to soil microbiology that are so important, but we don't often get the it's not as kind of clickable as say a
Yeah.
coronavirus paper. So it is,
Yeah, totally.
yeah. It doesn't
And
like
again,
get,
like, yeah,
yeah,
because the other thing is like this is just the description of that slower growth, but how it
mm,
impacts, right? That's going to take some other studies that are applying it. And like it took 15 years to get this piece of information, right? To like right? Slice through all that time,
yeah,
really, I think. And yeah, it's not an easy thing to extract this information, but it helps us build that hypothesis to how things will change, and then hopefully that will help inform people's strategies on how to manage ecosystems better.
yeah.
Okay, so
Yeah.
that's what we have for you this week. Join us next week for more cool microbiology news.
Yeah, we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. Science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
I totally agree. You can reach out to us over Twitter at microtwjc. We both believe that peer review is a process that we can all participate in, so that we hope you've had fun just hanging out with us and listening to us ramble about microbiology today. If you think you've had something to add or found something unclear, just let us know in the comments.
It's been a pleasure chatting with you, Danny.
Same here, Fuzz.
Tune in next week for more microbiology content, but for now, it's bye.
See ya.